Now, in a perfect world, our enzymes would do their job and they would know when to stop, they would know when to start, but that's not the case. So we have ways in which to check the working of our enzymes, but also ways in which we can combat enzymes of, let's say, bacteria. And that's how antibiotics can interact with them. These are called enzyme inhibitors. They inhibit the enzymes from doing their job. Therefore, they can no longer serve as catalysts within their biochemical process. There are two categories of inhibitors, reversible and irreversible. Looking at our reversible enzyme inhibitors first. A reversible inhibitor means that the enzyme is not going to be shut down permanently. It's only going to be temporary shutdown. We have these in our body so that enzymes don't keep just moving the chemical reaction forward when we have too much product being made. So there's this feedback loop that our body uses in addition with our inhibitors, and so that's also called allosteric control for positive negative feedback. All these fancy terms I'm sure you've heard at some point. Two types of reversible inhibitors. Looking at our competitive inhibitors first. A competitive inhibitor means that the substrate and whatever compound the inhibitor is can compete for the same active site. That means they need to have a similar enough shape that that induced fit model is working in its favor, but it's not getting the right R group interactions within the active site to do its job effectively. Looking at our image, here we have this enzyme that works on succinate. Succinate is part of the citric acid cycle. It's one of those intermediate compounds. And it's going to fit in there kind of like a puzzle piece, right? And you can see how it fits. What occurs from this succinate is it actually forms that double bond. So we remove a hydrogen from each one, forming that double bond, creating fumarate. Malonate is its competitive inhibitor. On malonate, I do not know if I'm saying these correctly, BT dubs, is different because it has one less CH2 than succinate, which means when it goes into the enzyme, it still has those carboxyl groups, so it still fits in there like a puzzle, but there's no way for it to remove hydrogens and form that double bonds. So this is a dehydrogenase, but it can't do that because it is missing it. Therefore, no product is formed. That enzyme cannot do its job, and fumarate cannot be formed and proceed forward. So this is a competitive inhibitor. Now, how we can get this enzyme back to functioning is we can flood the system or build up the system to increase the substrate concentration, and that can help displace any of the inhibitors present. Where this can occur is through a process like our citric acid cycle, where we have substrate 1, substrate 2, substrate 3, substrate 4, and we are moving through, and we get to a point where substrate 4, we have a high enough concentration. We can look at this through the citric acid cycle. You could look at this through glycolysis. If we have enough pyruvate, right, we want to go and stop that first reaction that happens. So once we have enough concentration of this substrate, we get what's called an end product inhibitor. It's going to go in and stop the function of that first reaction. So this picture is kind of an example of how that process could occur. Then we have our non-competitive inhibitors. Our non-competitive inhibitors are not going to resemble in shape that of the substrate, which means it's not going to interact on the active site. Remember, even though we use these cartoon style drawings, that enzymes and proteins have a lot of gaps. They're not solid. So another compound can enter anywhere in that space of the enzyme and be considered a non-competitive inhibitor. Here we have this inhibitor. It's going to go into this other site on the enzyme. When it goes into that other site, it's going to create that change at that site, and it forces all of like the enzyme, it's kind of like a cascading effect that ultimately changes the shape of the active site. Now the substrate no longer fits in there, and the enzyme cannot do its job. Lastly, our irreversible inhibitors. So these are going to be compounds that are going to permanently disable the enzyme from doing its job. This is where we really focus a lot of our antibiotics on. Super cool. Here we have penicillin. So penicillin will inhibit the bacterial transpeptidases. What that means is the transpeptidases is what catalyzes the formation of peptide bonds that need to be used for cell wall building. What penicillin has in it is it's called a beta-lactam ring. So this is a heterocyclic amide ring, and our atoms do not like to be at that 90 degrees, so it's a very reactive site. So it's that site, that amide bond, that is going to interact with that hydroxyl group on the enzyme we are functioning, and that creates a permanent bond. That enzyme is no longer functional to help build the peptide bonds necessary to build the bacterial cell wall. So we're stopping the function of the cell wall, so then the bacteria cannot grow and reproduce. Really, really, really neat process for a drug. However, bacteria are smart, and they find their way around it. So they have an enzyme that disables penicillin from doing its job. I love it. Right? So here we have that penicillin, we have that beta-lactam ring, but they have this lactamase. So it works on the lactam ring. And it disables it just like we had seen with the role of that penicillin's job for the enzyme for the cell wall. So the bacteria are like, don't think so. I'm going to disable you first. So then we have a way to disable those enzymes as well. It's like this back and forth game. The bacteria think we're going to get them, and they're going to get us back. And my money's on the bacteria winning ultimately, but that's okay. So again, we can have these points where we can interact with that lactamase and activating the lactamase from doing its job, so that then the penicillin can go back and do its work. So this is what pharmaceuticals, antibiotics, antivirals, this is what they do. They have to figure out what can be targeted in the construction of or generation of these bacterial or viral particles. What enzymes or proteins, their nucleic acids, what can we target to prevent forming, allowing it to work so that it cannot replicate and spread. And so it's an incredibly amazing process, but it's also why it takes so long to make antibiotics and drugs, because we are working with living things that can, as we saw in the second one, develop a resistance. So this resistance comes from that bacterial process of, hey, I'm going to disable your compound before you can disable us. So it's constantly this back and forth interaction that's occurring between the changes occurring in these single-celled organisms or viruses and what we can do to counteract them. Love this stuff. It is so fascinating.